Hello, everyone. So this week we're going to be discussing our last class of biomolecules, and those are nucleic acids. And we're going to talk a little bit about protein synthesis as well. All right, so let's dive in with the introduction to nucleotides and nucleic acids. So first of all, just take a step back. Nucleic acids are what make up both DNA and RNA. Okay, so that's going to be the main focus of this week's lecture, our DNA and RNA. But just to sort of take a step back, big picture here, these are all key players in your biochemistry along with proteins. This makes up the central dogma of biochemistry. Okay, so DNA is what houses all the information as to how to run a cell. DNA will replicate itself to sort of make multiple copies to, so when a cell divides or something, each cell has all that DNA, all that information. DNA is transcribed into RNA. Okay, so this is a process known as transcription. And then RNA is translated into protein structures. Okay, so all the proteins in your body, the code for them lives in your DNA. The DNA is first transcribed into RNA. That RNA is moved out of the nucleus in the cell and then translated into the actual protein structure. Okay, so kind of how I think about it is DNA is the information, it's where all that information lives. The proteins on the other end of the spectrum, the proteins, they're like the worker bees of the cell. They're what actually is breaking up the sugars and transporting ions across a membrane and all that good stuff. And RNA is the middleman between those. It's how the DNA information makes its way out of the nucleus to be translated into the protein structure. And so that's what this is sort of what we're going to be talking about in more detail throughout these next lectures. But I think just having the bigger picture here is important, right? We also talked about sugars and fats. Those are kind of energy sources, or they're the intermediate or the substrates of chemical reactions in your body. These are what are kind of doing the work here. Again, the DNA holds the information. The proteins are the actual little machines that are doing the work, and the RNA is the middleman that lets that information from the DNA be translated into protein structure. Okay, so first, before we even get into all that, we just need to take a step back and talk about like the structure of all of these things. Okay, so actually nucleic acids refers to the big long strands of DNA, and those are made up of little building blocks, much like our amino acids, but they're called nucleotides. And so that's going to be our first lecture here, is just talking about those individual building blocks, and then we'll put them together in a big chain in the next lecture. Okay, so those building blocks in both DNA and RNA, they have the same three components. One is what we're going to call a base, and we'll see what our different bases are for our nucleotides. But this is what differentiates one nucleotide from another nucleotide. So much like we had those R groups on our amino acids, and that's what made one amino acid like glycine different from another one like arginine was the identity of that R group. That's going to be what our bases do here, is they differentiate those different nucleotides. The other building blocks are shared by all of them. Those bases are linked to a sugar, okay, specifically to the anomeric carbon or carbon 1 on that sugar. And then there is a phosphate group that is linked to carbon 5 of that sugar. Okay, so all nucleotides contain these same three components. Okay. Yeah, so there are two types of nucleotides, the DNA and the RNA, and we'll talk about the differences between them here in a moment. Again, these can be linked together to form nucleic acids. We're going to focus first on those individual building blocks, which are called nucleotides. Each nucleotide contains the same three components. The base, this is what differentiates them. Differentiates nucleotides. There's a 5-carbon sugar and then a phosphate group. Okay, so those are what our individual building blocks contain. Again, the nucleic acids, those are the larger molecules when you start building those linking nucleotides together, and we'll talk a lot more about those in the next lecture. Okay, so let's talk about the different bases for now, all right? These are all heterocyclic amines, and they're all derived from the same two bases, either pyrimidine or purine. You don't have to worry about memorizing structures here, but we do want to be able to classify a nucleotide as either being a pyrimidine or a purine base. So let's just take a look at these real quick. This is our list of all the different bases that exist. Okay. The pyrimidine derivatives all have just one six-membered ring. Again, these are heterocyclic amine rings. So they're six-membered rings that contain nitrogens. In this case, there's two nitrogens. And the purines are also heterocyclic amines, but they have now a six-membered ring fused to a five-membered ring. Okay, so if we look within a class, we can see that they differ a little bit. So for example, cytosine has an amine group, whereas uracil and thymine have this carbonyl oxygen. So they do differ slightly, but nonetheless, you can see how they're derived from the same base compound. Okay, same thing with our purines. Adenine has a nitro group or an amine group, where guanine has a carbonyl oxygen. So they do differ slightly, but not crazy significant. Okay, so these are the identities of our five different bases. And right off the bat, I want you guys to sort of link these two together in your mind, uracil and thymine. Okay. Uracil is all the other nucleotides are found in both RNA and DNA. Uracil is specific to RNA, whereas thymine is specific to DNA. And you see that structurally, the only difference between these two is that thymine has a methyl group here. Okay, so they're going to play very similar roles. Just one's going to play that role in DNA, and the other one's going to play it in RNA. Okay. Again, the only difference structurally is the presence of this methyl group here. Yeah, I mean, I guess just sort of why the body has, why does all the other nucleotides are the same in DNA and RNA, but thymine and uracil are different? It actually has to do with the fact that RNA is very short-lived within the cell, whereas DNA is going to be there forever. And you don't want to have any mutations in your DNA. And it turns out that, long story short, by methylating this group here, nature found a way of preserving or decreasing the amount of mutations that would occur in the DNA strand itself. So actually, it's thought that all life was based on uracil back in the day, and then our bodies have evolved to have this methylated component, this thymine, because it prevents mutation in that long storage of DNA. Okay. Yeah, that's just sort of FYI. I think it's interesting, but bottom line is cytosine, adenine, and guanine are the same in both DNA and RNA, whereas uracil is uracil is only in RNA, and thymine is only in DNA. Okay. And we want to be able to classify these. So if I ask you, what is cytosine, a pyrimidine or a purine, you want to be able to know that. You could just sort of notice that the longer name leads to the shorter structure, right? So pyrimidine is a much longer word than purine, and pyrimidines are smaller structures, right? So the longer name leads to the smaller structure, and the shorter name leads to the larger structure. That's kind of just how I have it in my brain, so that it's easier to memorize. Okay, and so again, in DNA, you'll have cytosine, thymine, adenine, and guanine. Whereas if you go into RNA, you have cytosine, uracil, adenine, and guanine. All right. Okay, so what is the difference between DNA and RNA? So first of all, we talked about the bases that are present in each one of these. The big difference between DNA and RNA is the sugar. Okay, so RNA, The R stands for ribose nucleic acids. And in DNA, the D stands for deoxyribose. All right, the difference between RNA and DNA is this oxygen right here on carbon 2 of the sugar. In RNA, that's a normal sugar. We're used to seeing OH groups on every carbon in a sugar. So RNA has a normal sugar that has that hydroxy group. DNA, that oxygen's been removed, right? So carbon 2 does not have an OH group on it. That's where this name comes from, deoxyribose, right? So ribose is just the normal old sugar. Deoxy means that you've removed an oxygen, okay? So that's where the deoxy comes from. That's how you can remember it, deoxy, removing that oxygen. Yeah, this single atom is the difference between an RNA and a DNA. Okay, everything else about this sugar is the same. You can look and convince yourself of that. The only difference is that DNA does not have an oxygen on carbon 2. All right. Okay, so the whole thing together is called a nucleotide. A nucleoside is just the base and the sugar together. And these will link up to form a nucleoside that has a beta nitrogen glycosidic bond. So let's just zoom in here. All right, so we're linking together this nitrogen on our base to the anomeric carbon. It's actually not the oxygen, it's the carbon itself. And that forms this beta N glycosidic bond. Okay, and all of this together is what we call a nucleoside. All right, so carbon 1 of the sugar is linked to the base. When we add the phosphate group on there, that doesn't change anything that goes on the back half here. But now we have a phosphate group linked to carbon 5 of our sugar. So that's a phosphoester linkage is what that's called, this right here. This is phosphoester. You can see that if it was a carbon, we'd just call it an ester, but it's a phosphorus, so it's a phosphoester. And this thing altogether is what's called the nucleotide. Okay, so let me actually do this. Oops. This thing right here is the base. It's what differentiates all the different nucleotides. This right here, the base plus the sugar, this is the nucleoside. And the whole thing together, including that phosphorus, is called the nucleotide. Okay, and the nucleotides, the whole thing, the thing containing all three of those components, that's what's going to be linked together to form our RNA and our DNA. All right, so all the different nucleotides of DNA and RNA are listed here. They're just all those different bases that I showed you, but linked onto their sugars, and then with that phosphorus as well on carbon 5. So they're always linked on carbon 1 of the sugar. The bases are always linked on carbon 1 of the sugar, and the phosphate groups are always on carbon 5 of the sugar. Okay, let me just sort of zoom in here, what this is saying. We'll talk a little bit more about the name, but I just wanted to point it out, right? So if this is what I'm looking at now, especially if I just get rid of this, this would be the component in RNA. How do I know that? Because there's an oxygen on this carbon, carbon 2. If it was DNA, then I would have to get rid of this oxygen and replace it with just a hydrogen. That would be the nucleotide that you would find in DNA, okay? If it's an RNA, it would be adenosine monophosphate, mono because there's only one phosphate. If it was the component in DNA, which is what I just drew, you would call it deoxyadenosine. Okay. For uridine, the uracil and the thymine. Notice how, because uridine is only found in RNA, you only see it in this picture with the OH group, whereas that thymine is only found in DNA, so you only see it here with just that hydrogen. Okay, so difference between DNA and RNA. So first of all, the bases, they all share the A, G, and C, but then DNA has thymine, RNA has uracil. The big difference is the sugars, right? DNA contains that deoxyribose where it's missing an oxygen. RNA is just the ribose. The nucleoside is when you take the base and the sugar together, and the nucleotide is the base, the sugar, and the phosphate. The nucleic acid is when we start linking together those nucleotides. Okay, for the naming, I'm not going to ask you to name these things from scratch. Let's just look here. There's sort of a dropping of the, you kind of change the ending, I don't want to focus too much on that. Let's just look though. Adenine becomes adenosine. If it's in DNA, it would be a deoxyadenosine. If it's in RNA, it's just adenosine. Whereas cytosine becomes cytidine or deoxycytidine. I'm not going to worry too much about those endings. What I think is really important is the 3-letter abbreviations. Okay, so these are the 3-letter abbreviations for our nucleotides. Let's just look, first of all, let's do it in steps here. So first of all, if it has a D out front, that means it is a deoxyribose or a component of DNA. If it isn't, if there's no little d out front, then it's a component of RNA, right? So a nucleotide in RNA would be AMP, whereas a nucleotide in DNA would be DAMP. The next letter is the identity of the base. So A for adenine, G for guanine, C for cytosine, and T for thymine, or U for uracil. So that's what that next letter is. The M stands for how many phosphate groups are on there. And so there can be 1 to 3 phosphate groups. When it's in a nucleic acid, there's only 1 phosphate group. But the cell uses the triphosphate and diphosphates for other things as well. So the M, let's just say, so there's AMP, which has 1 phosphate, 1PO4. There's a DP, which has 2 phosphate groups, the di, and then ATP, or triphosphate, that has 3 phosphate groups. Okay, so being able to recognize that middle letter represents how many phosphate groups are, and then they always end in P. They also have that same P ending. Okay, so these components that we talked about, if there's a D out front, that means that it's a component of DNA. The next letter tells you what the identity of the base is, and then the middle letter, the next letter tells you how many phosphate groups are bound. Okay. Oh, so here we go. So here's an example of AMP versus ADP versus ATP. So here in AMP, I have 1 phosphate group bound. When I get to ADP, I have 2 phosphate groups linked together. And finally, ATP, I have 3 phosphate groups linked together. Okay, so that's what that middle letter abbreviation tells you. Again, in making DNA or RNA, it's always going to be those monophosphates, but the cell uses ATP and ADP a lot for other things. Okay, so give the name and abbreviation. Again, you know what? I'm not going to care if you guys can name it. I just care that you can be able to find the abbreviation for it of this structure here. Okay, so we'll do first the RNA version, where it has that oxygen on carbon 2. So what are we going to do? We're going to look up the base identity. And so this is going to be an intro into what you'll see on your constants page for this chapter. Ignore that for now. This is what you'll be given on your constants page. It's just a list of these bases in alphabetical order. First of all, you want to be able to tell me whether they are pyrimidine or purine derivative bases. You want to be able to tell me whether they're components of DNA or RNA or both. And then just be able to recognize them, right? So again, let's go back to our page here. I can tell right off the bat that it's going to be a purine derivative because it's the larger of the two structures. It's got the 6-membered ring combined with the 5-membered ring. And then the giveaway is going to be this carbonyl group right here. So when I go, that is the differentiation between guanine and adenine. So this would be G. Again, that's the RNA component, so I'm not going to put that little D out front. There's only one phosphate group on here, so this is a GM for mono, GMP. Okay, if there were more than one, if there were 2 phosphate groups, it would be GDP. If there were 3 phosphate groups, it would be GTP. There's only one, so that's how it's GMP. Okay, and then lastly, just to do kind of another example, what if I, instead of giving you this particular sugar, I had given you this? Well, now I know because that is the deoxyribose, because it's missing that oxygen, I will need to put that little D out front. Doesn't change the identity of the base. The base is still G, it's still MP. So this would now be DGMP. All right. I don't really care if you know the name or the trick about switching the endings. What I care about is that you know the 3-letter abbreviations. Okay. There will be some mastering chemistry problems that ask you for the name. For that, I totally am expecting you to use this cheat sheet here. For the other ones where it's just asking you for those 3-letter abbreviations, I want you to get in the habit of just using the constants page. All right. Whoops. So, yep.